So let's just walk through this Ecological Interactions Lab together and make sure that we understand what we should be seeing as we walk through the steps of the lab. So in experiment 1, I want to make sure that as you're viewing this video that you have printed off or have digitally available the procedure for this particular experiment. So for experiment 1, note that we really don't need very many supplies. We need vegetable oil, water, a 10 and 100 milliliter graduated cylinder. So step 1 tells us to measure 9 milliliters of water into the 10 milliliter graduated cylinder, which you see pictured here, and then to add 1 milliliter of oil. So I know it's a little bit difficult to see. However, you should be able to see the meniscus that shows where the milliliter of oil is, right here at this portion of the graduated cylinder. Now the next step in the procedure, if you're following along, asks you to record the volume of oil in the 10 ml graduated cylinder in table 2. So that is a table provided along with the procedure sheet. So the volume of oil would just be the 1 milliliter that we added. Now we are asked to pour that entire content, so the entire 10 milliliters, which would be 9 milliliters of water and 1 milliliter of oil, into the larger 100 milliliter graduated cylinder. So I did that, but then it asks us to repeat this 4 more times. So at the end of the experiment, this is the 100 milliliter graduated cylinder that you see pictured here. And again, I think you can see that there's a division about right here between the oil and water. Maybe a little bit higher than that, but we have approximately 36 milliliters of water and approximately the 4 milliliters of oil, right? Because I did this 4 times. So it would have been 1 milliliter of oil times 4 and 9 milliliters of water times 4. Now, one thing that I want to point out, so you'll also record this volume in table 2. But what I want to point out to you is, in organisms, we are mostly water, right? Animals are a high percentage of our bodies are made up of water. And we are constantly losing water, and therefore we have to take in water. So we lose water from our evaporation, through our respiratory system, from urine and feces. So the water is going to be something that is used by the organism, right? And is going to be taken in and leaving. However, the oil in this experiment is actually representing something that is not going to be lost in the feces or the urine or the evaporation. So it's something that's going to accumulate in the organism. So an example, if we set this out, this particular graduated cylinder, and we allowed evaporation to occur over several days, the oil itself is not going to evaporate, but the water, a significant portion of the water is. And then what we would see is over time, right, that oil is going to continue to accumulate in an organism. So as smaller organisms are consumed by larger organisms, we see that this accumulation of oil, or in the case that we're looking at, maybe a heavy metal or some toxic chemical, gets greater in the higher trophic levels, in those top carnivores, Now we're going to move to experiment 2. So if you would please get out the protocol or the procedure for experiment 2 and follow along with me. Now in this experiment, we needed quite a few more supplies, but they're all listed there for you. Majority of those are supplied in your kit, but there's a few others that we had to supply ourselves, like cooking oil, scissors, and liquid dish detergent. You'll notice that there's a baggie on the bottom left, and as you may notice in your kit, some of the contents may be loose, but sometimes you'll find baggies of multiple components. Now, if we follow through, the first thing we're to do is to cut a chamois and the 3 feathers that were supplied into 6 different pieces. So as you see here, this is just the chamois that's cut and the feather that's cut into 6 individual pieces. Now, one other thing that was important for us to do is to set aside a control of the chamois and the feather that aren't going to get any treatment. And the reason we're doing this is we want to be able to compare the chamois and the feather in their original state to our different treatment groups. So then if you follow along, we have 5 different beakers that we're going to have as our treatment groups. So we have one that we add liquid dish detergent to, one that we added powdered dish detergent, one that we added powdered laundry detergent, one that was an oil and water mix, and one that was just water. Now, the liquid dish detergent, powdered dish detergent, and powdered laundry detergent were mixed with water so that we could create a solution of those detergents. So that you can read through steps 5, 6, 7, 8 for setting up those specific treatment groups. Now, in step 9 is when we make sure we set aside the chamois and the feather as our control that aren't going to get exposed to any treatment. And then in step 10, we're adding a piece of chamois to the water, leaving it for 3 minutes, and then removing it and allowing it to dry on a paper towel. If you notice, step 11 through 14 are repeating that for each of the different treatment groups. So in other words, each of these 5 groups is going to get a piece of chamois that incubates for 3 minutes in the beaker, and then we take it out and place it on a paper towel that's labeled with the name of that treatment group. Once we've done that, we go through the same process, but this time using a feather in each of the 5 treatment groups. And ultimately, as you see here, we have a chamois in each treatment group and a feather in each treatment group.